are a family occasion, but not just only for Simon and his immediate family, but also for the rest of us, the bioengineering departmental family and the college community. We celebrate and acknowledge one of our own who has achieved the title of professor, and as such has recognition as a research leader in their field of endeavor. So Simon came to Imperial in 2004 as a lecturer, having completed 2 postdocs at Howard Hughes Medical Institute and UCL, following his undergraduate and master's degrees in Australia, in Monash and Sydney. He did his PhD at Oxford, and since coming here, he has progressed through the ranks to be appointed professor last year. Today, Simon will tell you a little bit about his research. He'll regale you with stories of his research excellence, and so it would be remiss of me not to mention some of his other contributions that are most important in an academic life. We know that academia is not solely about research. In fact, it is this wonderful balance, this synergy between creating new knowledge, communicating that new knowledge, and translating that new knowledge for societal benefit. And I think Simon epitomizes all of that. And so specifically, I just want to say publicly here that Simon is an extremely valued member of the Department of Bioengineering. When he joined us, we had 12 academic staff, we were a baby department, and 145 students. Now we have 46 academic staff and over 700 taught courses students. And this has been achieved through the hard work of a number of key individuals who have dug in, rolled up their sleeves, and helped to secure the sustainability and future of this discipline at Imperial College. And during that period of growth, Simon has made major contributions across the board. In teaching leadership, he was postgraduate tutor, then Deputy Director of Postgraduate Studies, then Director of Postgraduate Studies from 2011 to 2014. In research leadership, by driving the neurotechnology strategy for the department, resulting in significant growth in the field, not just in this department, across the college, many new appointments, international recognition of excellence in the domain for the institution, and perhaps even more significantly, leading the development, submission, and running of a doctoral training program in neurotechnology on behalf of the college. And also in departmental leadership. He was a member of the department's management committee from 2011 to 14, and he's such a valued member that we got him back last year, and he's back on departmental management. So Simon's an all-rounder. He has experienced the growth pains of being part of a nascent discipline and has plowed on through that in his own inimitable Australian way, pretty direct, and to make great personal achievements and contribute and support the achievements of many others. So Simon, I'd like to thank you on behalf of the department. On behalf of the college, we're proud to call you colleague and friend, and we are really looking forward to your inaugural lecture.
Thank you, Anthony. So, cracking the neural code and disentangling the wiring diagram. It's going to be the topic of my lecture, an engineer's view of the brain. And actually, I did start out very much as an engineer, looking back. But you know, we have choices in life. You come across intersections where you might choose one direction or another. This is actually one I came across a few weeks ago while off on my bike in Western Australia, actually, as it happens. Needless to say, I took Brainy Cutoff, the one with the little snake symbols. Much more enticing, isn't it? I faced also a decision point, back in my early days in training as an engineer. I actually started off doing microelectronics, designing chips. This was the first chip I designed. It was a silicon integrated fire neuron. You can see the transistors here. I actually fabricated it and tested that it worked. It's a little sort of micrograph over there. This was back in 1994 as a master's student. Inspired by that, I went on to look at, well, if I could make a neuron, maybe I could make synapses as well. This was my little sort of synapse circuit encapsulating what I'd managed to read from the neuroscience books that I'd found about how neurons and synapses worked. And I made one of those as well. It had lots of transistors in, as you can see. It actually worked. This is something that's known in the field as spike timing dependent plasticity. I made a little electronic circuit that could do that. So this was sort of back in my days as an engineer, and at that point, really, I started reading quite a bit of neuroscience and decided, well, actually, neuroscience was what I wanted to apply my engineering skills to. So rather than making more chips, I then left Australia. I came over to Oxford to study computational neuroscience, and one thing led to another, and here I am. So, okay, but an engineer's view of a complicated circuit, such as the brain. This is a complicated circuit. It's not as complex as the brain. This is the most complex thing, I think, that Intel have produced to date, or at least the most complex one on the market. And it's actually interesting to sort of compare where integrated circuit technology is with where brain circuit technology is in a number of facets. So if we compare this, how does the Intel Core i9 chip compare to the human brain, for instance? So here we have the chip on the left, a brain on the right. Transistor count, 7 by 10 to the 9 transistors. So quite a few transistors in the Intel chip here. Each of these is about 14 nanometers in size, and the power consumption of one of these chips is 140 watts, so about 20 nanowatt per transistor. How does the human brain compare to that? Okay, well, we don't have transistors in the human brain. We have neurons, so there are 100 billion of those. If you're comparing transistors to neurons, we're actually not so far away. Another order of magnitude and we're there. But actually, a neuron is a much more complicated thing than a transistor, and really the thing to compare would actually be the ion channel. And we have a lot of those in every neuron. We've been discussing how many that is. It's at least 50,000 and maybe as many as 1 million. So there are about 5 quadrillion or maybe even 50 quadrillion of these in a human brain. So actually, we're not there yet in terms of the complexity. Neurons are 10 microns, but actually ion channels are about 10 nanometers. So the chip technologists are actually getting there in terms of sort of ion channels, in terms of the size. But power consumption of the human brain is about 20 watts. So there's actually less than 4 femtowatts, perhaps much less than 4 femtowatts per ion channel. So really, this technology is much more impressive, and you can immediately see it's three-dimensional. It's got vasculature. There's all of these things associated with the technology that integrated circuit designers don't know how to do. So it is a much more impressive technology. And really, the job I sort of set my aim at completing is trying to reverse engineer the functionality of this circuit, but taking engineering principles to do so. So actually, just to play a little role-playing game, if we were trying to reverse engineer the functional properties of this chip, but we used the kind of techniques that systems neuroscientists were using for many decades, how would we go about it? Well, actually, the early systems neuroscientists what they're effectively doing was they're inserting electrodes into the brain, monitoring neurons, the first ones they came across generally, in the brain, and then trying to infer how the brain worked. Well, this is sort of equivalent in the chip to sort of drawing lines across the chip, measuring the voltage at random points on those lines, and trying to infer how it worked from that. So that's actually not going to be very productive. We need to do better than that. More modern neuroscience has produced things like this. This is the NeuroPixel electrode. It's basically a grid of densely packed electrodes, so we could monitor electrical signals of many neurons. If you were trying to use this kind of approach to the chip, well, it'd be a bit better, but it would actually still be very difficult to work out what's going on, because you wouldn't necessarily be looking at the right elements, the correct grouping corresponding to the architecture of the chip. And of course, brain circuits are actually very complex. So this is This is a rainbow image of the cerebral cortex. So it's basically obtained by randomly expressing fluorescent proteins of different colors in neurons, so that you get this sort of tapestry of the connectivity of the cortex. And you can see this sort of densely interwoven connectivity behind the cell bodies there. That's all the sort of the axonal connections and the dendrites internestled there. So it's actually a tremendously complex wiring diagram. And even if you are trying to infer the functionality of this chip, this isn't the diagram you need. That's just a picture. That's a micrograph, a photograph of the chip. It's this kind of picture that you need. So this is actually a much simpler computer architecture. This sort of describes the architecture of a very basic central processing unit. And you've got structures like arithmetic logic units and memories and registers and so forth, and the connectivity between them. We don't have the equivalent diagram for the brain. If we did, our life would obviously be a lot easier. And really what we need to do is to try and figure out this kind of wiring diagram and to monitor the signals amongst it. So it's actually quite a challenge. But I'm an engineer, and engineers build tools. Okay, so what I'm going to be telling you about today is some of the tools that we've built. I've taken a sort of a selection of the stories, I guess, that we've worked on in my lab. There have been quite a few other things that we've worked on as well. So I guess with apology to some of the people I've worked on on those things, I've selected just a few of them that I think sort of slot into this particular story today. One of them is the two-photon targeted patching robot, something we developed recently. Another is another tool from engineering, information theory, and how we've applied it to trying to understand how the signals in the brain represent information. Some imaging tools. How can we image brain activity during formation of a memory? Going back to that chip, I mean, really what you want to do is to sort of have that functional architecture, but actually watch the signals going from one place to another in the chip. So can we do the equivalent of that? Then finally, coming back to that wiring diagram, how do we go from a spaghetti-like wiring diagram, the kind of thing we saw with that rainbow diagram, to an actual understanding of the functionality of that circuit? Okay, so the key thing that we need to do in this, I would argue, is to be able to target circuit elements. So how do you go about targeting circuit elements? Well, you could do it by the function of the circuit. So in our CPU, that might be saying, well, select the arithmetic logic unit, please, and then we would maybe put our electrode into the equivalent of the arithmetic logic unit in the brain and sort of sample signals from it, or focus our microscope on it and sample signals from it optically. So targeting by function is one way to target, and it's an important thing to do. But we also probably need to target by connection. So another question to ask would be, what does the arithmetic logic unit tell the memory chip? And that's the kind of question we need to ask in the nervous system as well. So equivalent there might be sort of asking, well, what does the thalamus tell the visual cortex? Or what messages does the visual cortex pass back to the thalamus? So we need to have the technology to ask those kind of questions as well. And I'm going to be talking about some tools which help us get at both of these things throughout the next half hour or so. So, okay, how do we do this targeting? Well, actually, the molecular biologists have given us a big helping hand here. This is a complicated slide, and it's really just to impress upon you the complexity of the toolkit that we've actually got available from this. So we can use genetic techniques that basically allow us to take one of a library of mice, in effect, that have Cre recombinase in case of some of these, for instance, expressed in particular cell classes. Okay, so we can take a mouse, order it, in fact, from Jackson Laboratories in the US, and you'll get something which has got just a particular class of neurons containing this substance Cre. You might then cross that with a reporter mouse, which will put in just those cells a fluorescent protein. a red one in this case, a green one in this case. Or actually, you might be able to put something even more interesting in there. It might be some kind of functional sensor which lets you get at, say, the calcium signals in the cell or the voltage across the membrane of the cell and so forth. So this toolbox, which has come from molecular biology, is really the first element in this sort of toolkit we need to start answering these questions. So what we can do is we can take these mice, we can make mice that will have something like green fluorescent protein that we can see over here in a particular class of cells that we're interested in. It might be, for instance, just the cells in some particular nucleus in the brain that we're interested in. So that helps us target. But we need more tools as well, okay? So this is fine, but we actually need to be able to watch these signals in a live functioning brain. And the next tool we need here is the two-photon microscope. So I'll just give you a brief explanation of how that works. So over here on the left, we've got standard fluorescence, okay? So what happens here, you've got a fluorophore. It might be an atom in a neuron in the brain, for instance. It absorbs light, okay? So basically some light, a photon is incident upon this atom, and it causes an electron to go up an energy level. Some short time later, that electron returns down to an energy level, down to the ground state, and another photon is given off. So in this case, a blue photon was absorbed, and a green photon was given off. So that's single photon fluorescence. Two-photon fluorescence says, in this case, that electron going up the energy level was triggered by the simultaneous or near simultaneous absorption of a pair of photons. So we've still got this green photon given off when it comes back down again, but we've actually got a pair of photons, each of which had about half of the amount of energy of this one over here. Now that's a much rarer event, simultaneously observing two photons, simultaneously absorbing two photons. So to make that happen, we actually need reasonably high energy pulsed lasers to have it happen often enough to be useful to us. So why is this particularly interesting compared to this? Well, this diagram shows it, I think, quite nicely. Here we can see single photon fluorescence occurring. So the key thing here is that now you're focusing the laser onto a particular point here, that bit in the middle there, but nevertheless, if you observe a photon, you don't know exactly where it came from. It could have come from a small region, sort of smeared out in the depth direction there. In contrast, with two-photon excitation, there's actually some nonlinear physics going on here, which means that what you observe there comes from a very tightly confined region of space. With optical wavelengths, it's sort of the order of a micron in each dimension. Now, the thing about that is it means we know exactly where that photon came from on a scale which is actually smaller than the spatial scale of a neuron or even of some subparts of neurons like sort of dendrites or axons. So we can use this to observe in real time what's going on in them. And we do. What we do is we take these points and we scan them around in the brain and use them to observe what's going on. It's a little schematic of the kind of apparatus we might use. We're typically doing this kind of experiment in mice or rats. So here's our little schematic here. We've usually got a craniotomy, so basically a small hole in the skull, which is drilled. In principle, we can actually image through the skull, but with less depth penetration. And we're using a laser to excite, to focus the light down into the brain here, through this objective lens into the brain. The light that's given off then comes back up here. It's reflected across here and then split into a green and a red channel, which we observe with photomultiplier tubes. And we're scanning this around in 2 or 3 dimensions so we can build up images of what's going on in the brain. So this can be done in vivo, in live animals, live functioning brains. Here's an example of that taken from our lab. taken in our lab. So here are 4 layer 5 pyramidal cells. They're actually from a transgenic mouse in which layer 5 pyramidal cells contain green fluorescent protein, and we're able to image them in a live brain. We can image down to, in this case, I think it's 800 microns or so in depth, while the animal is alive. We actually image it, I should say, just one plane at a time. So these stacks of images take about a minute or so to collect, and then we can build them. But on a single frame, we can actually scan back and forth in one location and build up images in one region very quickly. Okay, so that's two-photon imaging. So we had one example before. We were able to put fluorophores in particular sets of cells of interest. Now we've got a tool which lets us look at them. Now, a third tool we need is one that lets us really monitor what's going on in the cells. And the gold standard, there's a few ways we can do that, but the gold standard way of doing this is called the patch clamp. And basically, the way this works is you insert a pipette into tissue, okay? You're coming down, and as the pipette comes close to a cell, you can monitor the impedance of the electrical impedance of the pipette, and you can see it starts rising as it comes close to the cell. When it gets really near the cell, you can provide a little pulse of suction there, or provide some suction and suck onto the side of the cell, and you get what's called a gigohm impedance seal. So you're sealing your pipette onto the membrane of the cell. Having done that, you can actually then provide a strong pulse of suction and really suck a little hole in the cell so that the contents of the pipette become contiguous with the interior of the cell. And you can use that to monitor pretty much everything that's going on in that cell, at least in the vicinity of the soma. You can also use it to modify the cell. You can put plasmids into the cell to sort of modify what's going on in there. You can put dyes in so you can see what's going on. So this is a very powerful technique. It was applied first in cell culture, then later in intact slice preparations, and then later still in vivo, so in a live brain. Now, when done in a live brain, it's a little more difficult than this. So here you can see these little sort of images of the pipette and the cell under what's called differential interference contrast optics. In a live brain, it's a bit more difficult because you actually can't see what's going on. You've just got to sort of insert the pipette and monitor the impedance and hope you get close to a cell, and then you might be lucky and sample the cell. So that's difficult, but now we can actually combine that with the two-photon microscope that I mentioned before, and that would be called two-photon targeted patching. So that was actually developed by a colleague of mine, Troy Margrie, some years ago. So this illustrates what's going on here. So here you've got a pipette filled with red dye. You can see under the two-photon microscope you've got a transgenic mouse where you've got a particular class of cells labeled in green, and the pipette has been moved down. You're putting some pulses in so you can monitor the impedance and so forth. And so under visual guidance, again, you can actually record from that genetically defined cell type. So it's genetically defined because of how we've labeled the green fluorescent protein into that particular cell. So that's great. You might say, well, actually, that makes it much easier. Well, actually, it is actually, in practice, harder than in vivo blind patching. Now, why is that? It's a little bit counterintuitive. The reason is because you actually care about the cells that you're collecting. If you're doing it blind, you will basically, you'll happily sample the first cell you come across, and that will be fine if what you're interested in sampling is layer 2-3 pyramidal cells, but otherwise you're not going to get very many of the cell that you're looking for. So you've actually got much fewer targets in this case. Now, the other problem with this is that there are extremely few experienced two-photon targeted patches worldwide. It's a difficult technique. Few people have learned this, and there's sufficiently few that they're not able to teach others and so forth. So it's a technique which has not really disseminated very widely. So really what we need to do is to increase the throughput of this. It's difficult, increase the reliability, so remove the human error, and actually even maybe more importantly, to lower the entry barrier to doing this so that the technique sort of disseminates further and is more widely useful. So how do we go about doing this? Well, the answer is robotic automation, okay? So not robots like this. This robot would be very bad at patching. I would not let this robot anywhere near our patching rig. But we need robots nevertheless, or robotic automation. And this is our robot. Okay, this is not our robot. This is Luca Anachino, who is a PhD student who worked on this. This is our robot. So basically, how does this work? I mean, again, it's a two-photon microscope, so you can see some familiar bits of the schematic there from before. In addition, we're controlling, we've got a sort of a robotic manipulator, 3-degree of freedom, or 3-axis micromanipulator, rather, which is controlling the pipette, which is advancing into the brain. We're monitoring the pressure, or controlling the pressure in that pipette as well, with a little electro-pneumatic regulator there. And this is all under the guidance of a computer. So what we wanted to do here is to really be able to scan around in 3 dimensions with our two-photon microscope, click on this cell and say, okay, I want to record this one now. So robot brings the pipette down and automatically records from that cell. And that's pretty much how it works, bar a few technical difficulties. So, okay, what you do, you basically place the pipette at the surface of the brain, acquire the pipette tip, you then select the location in the brain of your target, so that will be down here, and then the robotic algorithm sort of brings the pipette down towards the target, sort of through the brain with some positive pressure so that detritus doesn't get collected in the pipette tip. Now, one thing I should mention here is a little more difficult than that, because actually, if you're moving a pipette through the brain, it's actually moving the brain tissue itself, so your target actually starts to move. And you need to correct for that. It's sort of a bit like a sort of guidance system for an intercontinental ballistic missile. You've got to sort of do a closed-loop tracking. And in fact, the algorithm we used in the end is actually very similar to the algorithms they developed for exactly that purpose. But then once you've sort of done that, you're sort of coming down here towards your target, and then there are sort of automatic algorithms that are employed to engage with the target cell, form a seal onto the target cell, a gigome seal, and then eventually blast the little hole in the membrane to go whole cell. So, this is how that works. And so you can see basically, so what's happening is while the pipette is advancing down through the brain, we're actually using our two-photon microscope. It basically uses that as a sort of the eyes onto the brain of the robot. It takes stacks around the target area and does some basic image processing to sort of collect these regions of interest and track them, so that as the tissue moves, the pipette's moving into the brain, the tissue's moving, but it works out what that movement is and then works out what it has to do to compensate for that. And you can see a trajectory here as the pipette's going down. You can see these little wiggles that basically it's compensating for the actual movement of the brain tissue and eventually ends up coming directly onto the target here. So this works quite nicely. In fact, here's an example. So this example is a GAD67 GFP cell. So it's just a particular class of inhibitory interneurons that you find in the brain. They express a neurotransmitter called GABA. That is for my dad, who was asking about GABA before. And so basically, we can select these cells so that's our target there, and automatically perform the recording. And then once we've done the recording, we can actually do sort of standard electrophysiological characterization that you see here. Okay, that's one recording. Does it work systematically? Yes, it does. We can do it over and over again. So here's a number of more recordings made from exactly these same type of inhibitory interneurons, which we selected as a sort of a proof of principle for this technique because it's scientifically interesting and also it's a widely available mouse. How well does this work? Well, we can compare our robot with an experienced human operator. So we obviously had to train that human operator to be experienced, but basically, for both the robot and the human, they can get a seal onto a cell about 50% of the time. That drops to about 20% if you increase the requirement to actually be able to go whole cell. So the times it's not working, it's typically because there's a little bit of detritus or something in that the pipette has picked up as it's going down. And the robot, the main advantage of the robot here, although I wouldn't argue it's a particularly strong point, is that it actually works more quickly. So at version 0, it's working already as well as a human. And the properties of the cells of the recordings you get are really just as good as those achieved by human operators. So these are just sort of the standard things that would be used to assess quality of recordings in electrophysiology. So the quality of recordings is similar for robots and experienced humans there. So okay, this is one tool in our arsenal that we've developed for probing targeted elements of the nervous system. The other thing we need to think about is actually what the information, where is the information? What is the neural code? So coming back to a fragment of our little CPU wiring diagram over here, if you were able to monitor the signals coming from one area to another, the ALU to the execution memory, for instance, you need to know actually how the information is represented in those signals. So with the analogy of a spike train here, for instance, so what's the neural code? Is the information contained, for instance, in the number of spikes fired in a short period of time? Is it the actual time of the spikes? Is it actually the pattern of spikes across different cells? So those are different neural codes that we can hypothesize and assess. So to do this, we developed tools coming from information theory, applying them to neuroscience to ask these kind of questions. So I'll just give a sort of a brief. This is my one equation. I was told that every equation reduces the audience by half. So the half of you want to leave now. Okay, go ahead. So what is this equation? This is the Shannon mutual information equation, and it's really asking the question, thinking about it in neuroscience terms. Let's say we have a stimulus and a response. So the stimulus might be something that a mouse sees. Okay, it might be the direction of a moving stimulus in front of the mouse. The response would be the thing we're measuring in the nervous system. So we're using our patch pipette, for instance, and monitoring action potentials in a particular cell. So it could be counting the action potentials or measuring their timing or so forth. So how much does knowing the stimulus tell us about what the response was? So we can use this equation to work that out. And really, just to explain it, the thing we measure in our experiment would be the joint distribution of responses and stimuli. So we might present a stimulus many times, observe the responses, and measure that joint distribution. And we can use this equation here, so PR, S log 2 PR, S over PR, PS. Now, there's a basic relationship from elementary probability which says if responses and stimuli were independent, if they were nothing to do with each other, then this joint distribution would actually be equal to the product of the marginals. It would be equal to PR times P of S. If that was the case, you have the same on the top and the bottom, so that would be 1, and log of 1 is 0. So in that case, there'd be no information. And really what the mutual information does is measures the extent of deviation from that case. So how much is the dependence between the responses and the stimuli? So you can see that with a nice example here. So here's an example of a stimulus. In this case, it's just a random number between -1 and 1, and a response which depends fairly strongly on that stimulus. And there's a little bit of noise as well. A classical measure to use to look at this would be the correlation coefficient. And in this case, there's a very high correlation coefficient of 0.98. And there's also a high information of 1.7. The units of information I should add are bits. Okay, so that's fine. Now, what about if there's more noise? This is basically the same. The signal in these two cases is exactly the same. So the relationship between the response and the stimuli is the same, but there's more noise. Okay, in this case, the correlation coefficient goes down and the information goes down. So far, we could have just used classical linear measures like correlation to actually see what's going on. Let's take another example, though. Here we've got a much more nonlinear relationship between the response and the stimulus. In this case, they're actually a sine relationship. Now, the correlation coefficient actually would tell you there's no relation between these things. There's no linear relationship between these two quantities because it's a nonlinear relationship. But mutual information actually gives you the full relationship. So this is an exact sort of exactly the same information quantity as over here. So this measure lets us look at the relationship between two quantities, often in our case a stimulus type variable and a neural response type variable. Then we can use that to probe the neural code in the nervous system by comparing what we get if we make different assumptions about what that neural code is. So here's an example. This is something that we did actually quite a few years ago, so in the early 2000s, but it just provides quite a nice example. We looked at the rat whisker system. Okay, so the rat has got these sort of whiskers which have a barrel representation, it's called, in the somatosensory cortex. And there's a one-to-one relationship between each whisker and this patch of cortex which relates to it. So Matthew Diamond's group in Italy did recordings from this, and they basically repeatedly stimulated each of these whiskers sort of many times, looked at the responses. So each of these is basically a collection of the spikes at a particular time after the stimulus onset. And in this particular case, this is one particular cell here, it responds a lot more to stimulation of this particular whisker and a bit to this one and not so much to the others. So if you use information theory to assess what's going on here, looking at the information against a particular time window at which we're looking at that response, then you can quantify the response in two ways. You can do it just by counting the spikes or actually by looking at the temporal pattern of those spikes. But it doesn't make a difference here to how much information you've got. And really, Occam's razor would say the spike count is actually the code in this case. But that was one cell. We looked at other cells. Here's another example where actually if you stimulated the different whiskers, you got responses from multiple whiskers, but they were coming through at different times here. And if you actually compared the codes here, you actually found that the spike timing code conveyed much more information than the spike count code in that circumstance. And in fact, overall, for a large population of cells, spike timing codes contained about 44% additional information than what you got beyond spike counts. So this really was pointing us to the conclusion that the timing of the action potentials is actually very important to information coding, at least in this sensory system. And we were able to break further down this information into different components, looking at the firing rates versus the correlations between the spikes. But we took that further, and really what we found was actually it was the time of the first spike that really conveyed the vast majority of that information. So we were able to sort of take these tools originally developed for communication theory many years back and really use them to sort of probe what was how information about a somatosensory stimulus, a touch stimulus, was encoded in the nervous system. And we found that really this sort of spike timing information was actually quite important in that case. Another example, coming back to two-photon imaging. So here's some example where we used the two-photon microscope. Instead of using electrodes to record signals, we used our microscope to record signals. We checked with electrodes what our signals were like. So we took a pipette, so basically similar technique to the two-photon targeted patching, but this was done manually. We take a pipette in, stick it next to a cell. We've got another one here. We've stuck it next to a cell. We can record the action potentials here. And then at the same time, we're monitoring the calcium signals in a particular area of tissue. So we're using, in this case, a calcium dye. So we can take movies, and the fluorescence at any instant in time corresponds to the local calcium. And in fact, when you have an action potential fired by a cell, there's a calcium influx into the cell. So that therefore, we can use calcium-dependent fluorescence as a way to monitor neural activity. So we did this, and we could use this to monitor cells across a whole sort of area of tissue, and found, so here are some example region here, where we can see a time series from the red cell here, from the green cell here, so this is the calcium fluorescence. Every time we got one of these little spikes, it corresponds to one or more action potentials being fired. And what we found, if you look at the correlation between the activity of these different cells, it falls off over some hundreds of microns distance. So the cells nearby each other are really firing synchronously together. And as you go further and further apart, the relationship between them starts to fall off. They start to become asynchronous, firing at different times. So we thought, well, actually, maybe this is actually used as a code. We can use this synchrony, maybe stimulus dependence of the synchrony, as a code that might be communicated in the nervous system. So we looked at that further. In fact, here what we're doing is we're looking at patterns. So these are 7 different cells here. This is from an area of the cerebellum. So the cerebellum, an area involved in sensoromotor control. And this is an area of the cerebellum which has got sensory signals related to particularly touch of the lip and things involved in chewing and so forth as well. So we looked at that, and we can build up these little words across space here. We're looking at patterns of activity across space in the brain and asking, well, actually, is there something special about these patterns of activity across space which tells us about what the stimulus was? And actually, it turned out it was maybe not that special. It was interesting, but not special. So here I'm plotting the information versus the number of cells that we looked at, and this is a typical region. So the black line here is the full pattern code. If what we did is we actually destroyed the identity of which neuron fired which action potential, so we're just pooling spikes across the area of tissue, you actually lose a fair bit of information. So it does matter which neuron an action potential comes from. But on the other hand, if all of those cells had actually been firing independently, you would have got even more information. So although there was this spontaneous synchrony occurring here, contrary to our initial hypothesis, it wasn't actually doing something special in terms of sensory coding. It actually was destroying information. And this was the picture over a large set of regions as well. And that's a neural coding scenario that I describe as a labeled line scenario, where you've got each cell is effectively doing something independent. And this synchrony is there, but it's maybe doing something else, something related to readout of information from motor signals, for instance, in particular. Okay, going on to the next tool. You might notice I've had these little tool icon here that shows where there's a particular tool that we've contributed. The thing we're really interested in here was looking at how neural activity occurs during a memory task. So if you're learning something new, so you're going to remember something new, or if you're recalling something that you've previously remembered, how does the activity differ? Now, this is actually a quite complex challenge in neuroscience terms, because we need to have an animal trained to do a memory task, and we need to image the activity in these cells while the animal is doing the memory task. But we were able to do this, and this is our preparation here. So we're looking in the hippocampus. It's a fairly deep area of the brain, which is known to be strongly involved in formation of new memories in particular. What we've done is we've delivered a virus into the hippocampus, which puts into the hippocampus something called GCAMP6, which is a genetically encodable calcium indicator. So once the neurons have been infected by that virus, they will express this calcium-dependent fluorophore. So that when we image with our two-photon microscope, the fluorescence we see will depend upon the calcium and therefore the activity of that cell. We also put in it something called mRuby, which is a red fluorescent protein, and that was a structural marker which just let us see where the cells are. And these are the kind of movies that you can actually that you can see in the brain. That's 13 times actual speed, so it's a little faster than what actually occurs. You see these flashes. These flashes correspond to action potentials being sort of transmitted amongst the neurons in this area. Now, the third thing we were able to put in there was, you see this purple channel here, a dye called methoxyo4. Now, methoxyo4 labels amyloid plaques. The other thing we were interested in here was looking at, well, how does this go wrong in Alzheimer's disease? And we've got mouse models of Alzheimer's that let us look at particularly the effects of these of these amyloid plaques on the local tissue. So that was another thing that we're sort of looking at there with that project. So we can do this during behavior. So that's just imaging, but now you can see we can actually do it during behavior. So I'll just explain the apparatus here. So the animal is under the microscope, so its head is fixed in one place. However, we're able to image by using this neat little platform. So there's actually, this platform here is actually, it's got a lot of holes in, hooked up to an air system. So there's air coming out of this, and this is a little carbon fiber platform there which actually floats frictionless on top. And the mouse can move around. And you can see example here. The mouse can move around, around and around, but it's actually the environment which moves. The mouse is actually in one place all the time so that we can image its brain. And you can see the trajectory of the animal as it's sort of going around here while we're under the microscope. This is the objective lens of the microscope. So normally what we've been doing here is we have sort of a circular linear track. One of the things we were particularly interested in was looking at what's called place fields. So there are certain cells in the hippocampus which respond when an animal is in a particular place, and we can use those as a readout of memory, of spatial memory. If the animal remembers where it is, then the sets of cells will respond according to its memory of that location. So we've used this sort of circular linear track, and we can use other environments as well. So typically our experiment works something like this. We might, over a week of training, have the animal learn two environments. We'll call familiar 1 and familiar 2. And then on an experiment day where we're doing imaging, we might put it in the environment familiar 1, and then we go to familiar 2. And that's an example of a memory recall process. We can image the activity of the hippocampus while the animal is recalling the spatial memory. Another experiment we can do is we have another stimulus, another environment rather, which we call NOV here, which the animal has never seen before until that day. So if we put the animal first in this familiar 1 environment and then go to the novel environment, this is an example of encoding a new memory. So here we were recalling a memory. Here we're actually laying down a new memory, and we can watch the neural activity as this happens. I'll skip over those for time. We had some more tools that we've developed in order to actually sort of automatically find the cells in the brain and pull out regions of interest. Now the nice thing, using this technique, we're actually able to map, able to look at the functional signals in the brain, not just on one day. So here's an example on one day here. These are the same cells going back on day 2 and going back on day 3. So we're able to come back to the same cells over time. And we're able to use this to watch spatial memories form. So here we've got 140 cells down on this axis of this plot here. And on this axis, this is how far we are around this circular linear track. Now, each of these cells, basically what I've done is I've plotted the activity averaged over many loops around the track. These cells that are so-called place cells, they're responding when the animal's at a particular point on that track. So you can see these little sort of groups of bright pixels there correspond to the activity of the cells when the animal is in its preferred, that cell's preferred location on the track. We've sorted them according to the bit of the track they like. So hence you can see this kind of this sort of diagonal line here. Now what happens if we take the animal out of that environment and put it into a new one? Now the new one in this case is actually just the same environment being run in reverse direction. Now I gave it away. Basically it gets completely scrambled. Okay, so each of these cells is no longer responding in it doesn't look like it's responding in a space-specific manner, but actually if you resort these cells according to, in the same sort of criteria here, you can actually see that there is. So what's actually happening here, we're watching a new memory being formed. So there's less of the cells are place sensitive, but some of the cells are starting to form this new place representation. So they're laying down this new memory. And we're sort of watching that process as it's happening. Now we're now able to actually record from the same cells for up to 2 weeks. So here's an example for up to 2 weeks. Day 14, I should emphasize, was actually yesterday. So this particular data is actually very recent. But we're excited about it. And so we're able to sort of watch these cells over several weeks. Now, during that time, the virus is continuing to sort of express protein in those cells. So the brain is not exactly the same, and we're still sort of working on the tools to really make sure we can track all the cells from day to day across all of that time. But we can certainly do it to some extent. So really, that work is sort of ongoing, and we're particularly looking at mouse models of Alzheimer's disease, focusing on something called the aberrant excitability hypothesis. So some cells basically near the plaques have decreased activity. Some cells further away from the plaques have increased activity. And really we're trying to look at how this actually affects ability to sort of lay down new memories and to recall old memories, and how that relates to the circuit activity there. So finally, I mentioned the wiring diagram, so disentangling the wiring diagram. So how are we going about that? Well, so really the job is to take this spaghetti and to turn it into something a bit more sensible. And to do that, we're using a two-photon microscope as well. So we just look at this diagram over here. What we can do is we can take the brains. It's actually the same brains that we've done those memory experiments in. At the end of the experiment, we kill the animal, and we take the brains and we sort of store them and process them. And then we can slice. We can image 100 micron thick sections, for instance, slice that off, image the next 100 micron thick section, and build up a three-dimensional picture of the entire mouse brain. So the example I'm about to show you is that for looking at an Alzheimer mouse model. It's called the 5XFAD mouse model. It's a model of early onset Alzheimer's disease. And this is, actually there's a lot of amyloid plaque in this brain. It's actually quite an old mouse for this particular mouse model. And you can see, so gray is actually the structural image of the brain, and purple is the amyloid plaques that you can see in this little fly-through. So that lets us really look at the distribution of functional markers throughout the whole brain. So in this case, it's amyloid. We can also look at mouse models, look at mice which are, for instance, labeling particular cell types. We're interested in a particular class of interneurons, for instance. This is another 3D representation, but it's a flat one. The color indicates the distance really into the picture here. And so this is a mouse that we were using in this in a particular study on the thalamus, and we're able to sort of see that this particular class of cells that we've labeled they're distributed in this case across a number of brain areas, but in particular the ones we're interested in looking at. Now, okay, but that's just cells. Really, the next thing we want to do is actually to be using this method to trace the connections. So this was the whole point about tracing connectivity. And for this, we use a new tool, the rabies virus. So basically what we do, it's a multi-stage process. So you inject two viruses. One of them delivers into the cell population of interest G protein and a red fluorescent protein so that you can see it. Then you inject another virus, which is actually a modified rabies virus, which has deleted the G protein. So this rabies virus can only replicate when it's put together with that G protein. So only in those cells that have previously been infected by this virus will that rabies virus be able to replicate. But once it goes into those cells, then it can replicate, and actually it can cross synapses. It can actually transmit back across presynaptic junctions. So it will actually label all of the cells that are talking to the one you've actually infected. But once it gets into those cells, there's no G protein around anymore. So it can't replicate any further. So it can cross exactly one synapse. So we can use that tool really to start tracing the connections of individual circuit elements. So here's an example, and it's really the final sort of real slide here, where we've actually injected this into the lateral geniculate nucleus and labeled a population of cells in the visual cortex which are projecting onto those cells that we've infected in the lateral geniculate nucleus. And there's two particular cells that we've traced here. You can see one in green and one in red. And you can see the sort of exquisite level of detail we actually get with this system for tracing the connectivity. I should emphasize that this is a collaboration with Steve Brickley's lab and a student, Gerald Moore, who's been working on this with us. So finally, I'd just like to end with taking that particular picture into the future. So that was great, but that was just using green fluorescent protein in this case. So we put a marker into the cells we were interested in, the cells that talked to the cells that we'd originally looked at, so we could label them, we could see what they were. But really what we want to know is actually what they're saying. What information are they providing to the cells we were looking at. So in this case, it's what information does the visual cortex provide to the inhibitory interneurons in the thalamus. So it's a sort of feedback information loop. So how do we do that? Well, actually, instead of using GFP, green fluorescent protein, we can substitute a functional marker like GCAMP, genetically encodable calcium indicator. In this case, the latest and greatest one is JGCAMP7, for instance. We could put that at some element in our circuit, so I'm going back to our computer architecture here, and it would transmit back to provide a functional label in just the cells that are talking to that one. And that really, I think, is the direction we need to pursue here, to map the functional signals from one place to another in the brain. That's how information is actually being processed and sent around the brain. So I'd like to end by thanking my research group, and I should emphasize this is actually more than just my research group now. Amanda Faust was originally a postdoc, is now a lecturer, and sort of building her own group, and we're sort of in a sort of partnership where she sort of leads on the optics side of things, and I lead on the systems neuroscience and engineering aspects. And many other members of our team, many alumni that I've previously worked with, and collaborators. And many of our sponsors here, and I particularly like to thank Mrs. Ann Uren and the Michael Uren Foundation, who funded the work on memory that you've just heard about there, and Alzheimer's. So thank you for your attention.
So at this time in the proceedings, we have a formal vote of thanks, and I'll just introduce the person giving the vote of thanks. So Bill Wisden holds the chair in molecular neuroscience at Imperial College, a fellow of the Academy of Medical Sciences. He's a collaborator of Simon's and was also instrumental in the establishment of the Center for Neurotechnology at Imperial College. Bill, the floor's yours.
Okay, thank you very much. So I'll try to do better than my wedding speech, which is why I just said thank you for coming. So I'll add a few extra sentences to that. So it's a great pleasure to give the vote of thanks to Professor Simon Schultz. And we've just seen and heard a beautiful blend of engineering, physics, maths, biology, and that's the Imperial approach. That's why we're one of the leading technological universities, because of people like Simon blending all of these disciplines together. And we've also, I feel, learned from his presentation the value of changing fields. I mean, as a biologist, I'm particularly impressed Simon does maths, but in general, he's a person that moved beyond his comfort zone, and I think, and also he moved halfway around the planet. So I feel these factors together, pushing the boundaries, is reflected in his science. I should also say Simon's actually a very modest person. So although he told us about one of the ambitious aims of his research, which is to crack the brain, to actually understand the brain, what he didn't tell us was that he's also one of the founders of a new discipline, which is neurotechnology, and this was briefly alluded to, but he's actually set up a graduate school for this here at Imperial, the Center for Neurotechnology, and this brings together scientists that are biologists, engineers, medical doctors, physicists, mathematicians, and I would just read from Simon's CV about the Center for Neurotechnology. So his aim, by founding this center, is to lead the development within biomedical engineering of the emerging field of neurotechnology, harnessing developments in photonics, information technology, materials and microelectronics to make advances in brain science and medicine. So that's the other half of Simon's work here at Imperial, which we didn't hear about, so I think he's been a bit modest about that. What else should I say? Well, as somebody that knows Simon over the last, I think, 10 years, I've always been impressed with Simon's enthusiasm, his high work rate, and his unflinching attention to detail. I don't know if that's, he's a sort of double thumbs up person. I always feel zapped up when I interact with Simon, so it's probably the Australian in him. But he's basically a very positive person, and I think that's another element in the success he's had with his research in tackling this very difficult problem of neural coding. And, you know, he is, although he did an amazing cycling holiday, which I followed on Instagram a few round, he is actually a workaholic. So I just counted from Simon's CV. He's supervised, he's not that old either, I don't think you're that old, Simon. He's supervised 26 PhD students. I mean, I find that quite amazing. And you've got 195 publications. So that's pretty impressive work rate. What else should I say? I know that many fans of Simon are here tonight. Family are here, father, wife. And also, you're a hugely collaborative person. So there's many of Simon's collaborators here. And I think Simon's a bit like a key node in the brain. It's like one of the key neurons in the circuits we just heard about. He's helping, as well as doing his own work, he's helping others do their work. And if he weren't here at the college, we'd all be at a loss. So we're very lucky to have Simon here at Imperial. So I'd like to wish you success in your future work. And there's drinks afterwards, I should say. But please join me in congratulating Simon on a wonderful lecture and becoming professor at Imperial.